So before you start, I just want to make sure that this tutorial has a optional image segmentation step, which is using a Giotto wrapper of cell pulse. And to use that, you probably need to um, create and install a different Giotto Python environment using this first chunk of code. If you are having issues with running this code, because we've noticing a few issues on this part, specifically for a MacBook. One way you can bypass that is to install the Anaconda from the Conda official website and can go to the Giotto GitHub page and find the Giotto environment or with cell pulse configuration and use this YAML file to create a new Giotto cell pulse environment. And that seems to be having less issue than the one just directly use reticulate here. And you can try to see if you successfully activate the Giotto cell pulse environment and run this package check to see if the installation is correct. So if it does not return anything, that means the installation is successfully. And before you activate any new Giotto, any new Python environment, you will need to restart the R session by typing .rs.restartR, because this is required by Reticulate if you want to switch Python environment for each R session. And also, please consider to download the file by running this chunk of code. Download the file for the Luna 4 example. And if you are having issue downloading file from the RStudio, you can just like copy and paste this link in your web browser and it will download it automatically. And you just need to remember to put it in the data structure, folder structure like data/day2 session 4, which will be need this to use for downstream analysis. So this tutorial is aimed at analyzing spatial proteomics, and more specifically, it's more or less about imaging-based spatial proteomics technologies. So it is compatible for a lot of different kinds of image-based data, such as Akoya, SciSif, IMC, MIBI, and Luna 4, which is sequential immunofluorescence. Please note that this tutorial focuses on starting directly with the intensity data, which is the image. And I know some of the platform will output the decoded count matrix, and that's not what we are, not this tutorial is for. So in short, for the immunofluorescence based multiplex technology, they use similar strategy, which is use a way to use the antibody to stain your tissue section, and you image after the staining. You take an image, and after that you elute the antibody from your tissue, and then restain for the next antibody. So that way you can multiplex more than like 3 or 4 antibodies at the same time, while you are while the usual for microscope only be able to capture a few channels of images. And this is the example data will look like. I select a few channels to show, which is a pretty nice picture that shows that you can multiplex different antibodies within on one section. And now we can go to the RStudio part. So basically, there could be different type of data to start with. One of the most common one is the OMI-TIFFs, because it is a sort of an extension format of TIFF, and would be able to store the metadata information as well as multiple channels and also probably multiple time elapse point data for this. And for that, we can directly read in the metadata, because this is what you will get directly from the Luna4 platform. You just use OMI-TIFF metadata, will return a data frame, data table of the channel number and the staining antibodies that is saved in the OMI-TIFF file. But sometimes if you crop, like manipulate, or just by simply cropping the OMI-TIFF not in a correct way, some information might get lost, especially for the metadata. So that way, we probably need to read in a separate XML file, which is stored in the original big OMI-TIFF file, and parse that all with XML2, and then you can get the same thing from the XML file. And some platforms may also deconvolute and output grayscale single channel images. And we can create the single channel images from OMI-TIFF so that we sort of start with the same format of data if the platform will provide grayscale images. So what we can do is to read in the Luna4 OMI-TIFF via Terra. And what we can do is to extract the first channel of the Terra raster and create a Giotto large image on that. And this is what you will see if you plot the single channel Giotto large image. And then we can simply just write a for loop to loop over all the channels and subset with Terra to extract each single channel layers, and then write to a folder that you give to the file name of Terra raster. Okay, so it's written. And now that we have a folder of single channel images, so it's going to be something like a single channel, and you have all the TIFF file, single channel TIFF files. Single channel TIFF files that just have the antibodies and tagged with TIFF at the end. And then what you can do is to create a Giotto large image list directly from this channel, this folder, and it will output a list of Giotto large images. And you can append the image name to the Giotto large image list and directly plot one of the single channel images out of that. So this is going to be, if you take a look at the momentum, the expression will look like this. So now we have a list of Giotto large images, which is two of the needed files, two of the needed objects you need to input to create a Giotto object. The other thing we need is the cell segmentation polygons. So cell segmentation is necessary to generate single cell level protein expression data. So there are multiple algorithms to generate segmentations from images directly, and output could be different. And for that purpose, Giotto provides several functions, including create Giotto polygons from mask, create Giotto polygon from data frame, and create Giotto polygon from GeoJSON to load different file format to the Giotto polygon class. So here we use example for the output from deep cell mesmer algorithm. So for this one, we actually create a pseudo membrane staining channel, an overlap between 3 different channels to create a pseudo membrane, and the result is actually looks like something like this. So this is created from different channels and overlay that to a single channel. And this can be used as input file for the deep cell mesmer. And what we can get out of the deep cell mesmer algorithm is a mask file. So we use the created Giotto polygon from mask to create this Giotto polygon class. But let's take a look at how it looks. So this is how the Giotto polygon created from the mask file will look. And we can also zoom in and just plotting the overlay between the DAPI channel and also the cell segmentation. And it's working quite well. Like in the middle, you see the nuclei, and this is the boundary of the cell. And if Giotto also provide a wrapper function for cell pose to perform the segmentation. So what we can do is to save some time, we can crop the Giotto large image into smaller region, probably with the same extension, same zoom in area, and write it to the disk. This will be used as the input for the cell pose segmentation function. So right now, the input for the cell pose segmentation will be you need to import a image directory, and also you need to provide a mask output, because it will write to your disk as your segmentation result. You will need to provide 2 channels. By default, it's going to be 2 zeros. It's going to be set the grayscale version of cell pose. And for the model name, currently we can only support the default like pre-trained cell pose models, which is cyto, cyto2, cyto3, and nuclei. You can load that with this cell pose function. You can also pass on all the cell pose input threshold, like flow threshold, cell prop threshold. The default is going to be the same as one set on by cell pose. And more importantly, there is a specific requirement for the batch size. So this is the sort of represents how many 224 by 204 ROIs to be calculated in parallel. So increasing this amount will increase the RAM requirements and virtual memory requirement if you're using a GPU. And lowering the amount will increase the runtime. And for more information, you can take a look at the cell pose website like this. So right now, Giotto only supports the pre-trained model, but in the future, we hope to support the user-defined model as well. So simply, you can do list files, directory, cell pose models. So if a user create a model and put it here, it will you can just specify the model name to custom or something, and then it will take the user-defined model and run the inference one for that. By that being said, the cell pose segmentation has been done, and we can plot the overlay by plot the Giotto image first, and then plot the Giotto polygon next by setting add equals to true and border equals to red. What we can see is that the segmentation for these nuclei probably needs some fine tuning, which is normal for most of the deep learning based self-segmentation algorithm. You probably need to fine tune the model for your customized generated data. And now we have both the Giotto image list and also the polygon information. And then we can create the Giotto object. We use the Giotto object subcellular as well. What we can see from the Giotto class is that it has subcellular information, the polygon itself. The aggregated information is not yet available because we haven't run the overlap of between raster image and the polygons. Get spatial locations, we get 21 attached images. We did not see the point information here because we did not import a Giotto points, just like what you will do for like in situ transcripts. So after that, we need to run the calculate overlap between, if you just specify the spatial info to a certain polygon, and also the image names to like the names within Giotto. For that purpose, we can actually use a show function to see what images are available in Giotto. Show that. So if you do show Giotto image names, and you'll get a list of names. And if you only want to calculate one gene, you can specify the names here. And then you need to run the overlap to matrix, so it actually create a count matrix and sets the type to intensity so that it will create the aggregate of intensity values from the image itself. Also specify feature info equals to protein. And this is the count matrix with the overlap to matrix will create. You have your spat unit equals to cell, and you have feature types is protein, raw values because you are just aggregating the intensities, and you have a count matrix of 21, which is the number of antibodies we have, and 4,000, which is a cell number we have. So note that we actually have a DAPI expression here. DAPI staining is usually used to just stain the nuclei, and usually the intensity value for the DAPI is not very super meaningful. And for that purpose, including DAPI and also some like platforms will use some reference channels to adjust signaling calling, such as TRIC or Sci5. And these channels actually needs to be removed from your count matrix for downstream analysis. So what we can do is to extract the count matrix using the get expression file function, write it to a matrix file because we expect that output to be a matrix, and then remove the DAPI line. And if you have TRIC or Sci5, it's also going to be removed here. And then you can use the set expression function provided here. It specifies protein name and also the new matrix you want to set. Note that you will need to create a new Giotto expression sub-object to overwrite the previous one. So as we can see that after the override, the count matrix become 20 instead of 21. Giotto also provides a quick manipulation of the polygon, so that way you can actually enlarge or make it smaller of a polygon. Like what I'm showing here is that you use this rescale polygon function, inputs the Giotto object, inputs the polygon information, and name means it's what type of what what's the name of this new polygon information will be. And fx and fy means you are rescaling the proportion of the polygons. You need to rescale at a certain axis. So if I specify that to 0.7, it's going to be scaled both ways to 70% of the original input polygons. And then you can use another getter function for this new polygon, but just by specify the polygon name equals to small cell, and plot that onto the same zooming the same zooming extent of the whole data set. You can plot the DAPI image first, and then plot the original polygon by using add equals to true, and use the small polygons to be again add equals to true, but set the border equals to red. And this is the result you will see if you run the manipulation of this polygon. And note that if you enter a value greater than 1, it's going to be enlarged of your polygon. And next, we have the count matrix. So we run the normalization and add statistics, which is just using the standard Giotto analysis pipeline. But for the normalization of protein purpose specifically, we need to know that it may require a different normalization method, such as what George just showed, like this Giotto image, and you import the Giotto, and then let's say GPT-4. You have you'll see the density plot of the expression intensity values, and you probably need to do some manual cutoff of your input image, and then go through this normalization method. The next step is just to run PCA on the Luna4 image, and you can do scree plot as well. You can also do run UMAP, create the nearest neighbor network. And also, since we only have a very limited number of total features, usually latent clustering or any other type of graph-based clustering method does not work as well as what we see in either single cell or VZM clustering. So here we use hierarchical clustering and just specify a vague number of clusters you would start with, and then you can run the spat in situ plot to plot the different polygons with the hierarchical cluster number. And to annotate the different clusters, there's a quick and easy way to do is that since we only have 20 protein markers, we can just plot all of them with a heat map. And just by looking at the expression of these heat map, we can actually give a quick annotation to the Giotto object. So like I create an annotation of B cells, macrophages, T cells, stromal, epithelial, DC cells, fibroblasts, and endothelial. And the names of the annotation needs to be in the same order, as the hierarchical cluster number. And then we can use the annotate Giotto function to actually annotate these hierarchical cluster into the cell types. And what you can do next is to create a spatial network. By default, it will create a delineary network, and you can sort of see what's connected, and what's considered to be a connection between each cell types, and you can see how the network is nested. And what you can do next is to you run the cell proximity test, which is simulate. Oh, haven't run. Oh, okay. So if you run the cell proximity test, it will run through some simulations and test whether or not this connection is considered to be a significant connection. And what we can do next is to plot the proximity bar plot and see what's enriched and what's depleted within different cell types. Like for the top ones, it's like showing some connections between within each cell types. And for the downstream like red bars shows the connections between different cell types. Another way to show this is actually to plot the proximity network. So here, what we can see is that these cells are closely connected to each other, and the non-significant connections of cell types will be labeled as with a different color scheme. I think this is the end of my part.